Okay, so for our module 6, amino acids, peptides, and proteins. So ito na yung part na magme-measure na tayo ng analytes from our samples, be it from blood or the urine. So amino acids are the major metabolic intermediates and the basic structural units of proteins. So yung proteins natin, binubuo siya nitong mga amino acids. Sila yung mga parang building blocks natin para magkaroon ng proteins. So the biochemistry of amino acids, they are organic compounds containing both an amino group, NH2, and the carboxyl group, COOH. So sa biochemistry niyo, ito yung mga napag-aralan niyo, yung structure, formation, and naming of our carboxyl groups, yung mga additional natin doon. So acid-base properties. So at neutral pH, they both have positively and negatively charged groups, and of course as zwitterions. So zwitterions yung tawag natin sa particles. Next, so isoelectric point. So the pH where the amino acid and other molecules have a net charge of 0. So yung tinatawag natin na isoelectric point. So they are water soluble and they are stable in the plasma. So metabolism, they serve as a substrate for protein synthesis, so sa formation nga ng ating mga proteins. So sources, pwede sa diet or endogenous turnover. So for breakdown, pwede rin makakuha ka ng mga amino acids. So daily requirement, 0.8 grams per kilogram body weight. So, per kilogram, kailangan meron kang 0.8 grams ng protein na kinakain or yung amino acid. So, they increased during growth and many disease state. So, mamaya makikita natin kung bakit or para sa yung alin yung mga nataas-taas na bilang ng ating proteins. Metabolism again. So, essential amino acids. Meron kasi amino acids na hindi kayang i-produce or they are not synthesized by the humans. Ito yung mga yon: isoleucine, leucine, lysine, methionine, phenylalanine, threonine, tryptophan, and valine. So they are the amino acids that cannot be synthesized by the humans. So amino acids are released from dietary proteins by degradation with pepsin in the acidic environment of the stomach and the pancreatic hydrolysis. So digestion of protein may be impaired by pancreatic or gastric disorders. So dito kasi nanggagaling yung mga enzymes natin pang breakdown ng protein para maging amino acids. Amino acids are actively transported by GGT or gamma glutamyl transferase and maintains higher concentration of amino acid inside the cells than outside. So mas maraming amino acid sa loob ng ating mga cells kaysa sa ating labas ng cells. So they also act as intermediates in metabolic pathway. So sa urea cycle, they converts ammonia to urea in the liver. Sa alanine cycle naman, metabolic pathway that allows muscle cells to use amino acids as fuel source and exerts excess nitrogen in the form of alanine. So the formation of urea and the formation of alanine. So they undergo efficient glomerular filtration but are reabsorbed in the proximal tubules in the kidneys. So pag healthy ang kidney natin, i-filter siya ng ating mga glomerulus pero nag-absorb ulit tayo sa proximal tubules. Kapag naman nagka-problema or sira ang ating kidneys, pwede kang magka-amino aciduria. So increased renal excretion of amino acids. So, ini-excrete natin siya in the urine sa halip na i-absorb ulit siya para sa ating katawan. So, this may be due to the overload or from tubular impairment. So, pwedeng overload kasi sobrang dami or sa tubular impairment, hindi na siya na-absorb ng ating proximal tubules. Premature neonates have a generalized amino aciduria. So, kapag premature ang baby, mataas ang kanyang amino acids sa urine. Specimen considerations. So, concentrations are high during the first days of life. So, mga bata, or baby, kapag mga baby pa, madami or mataas ang concentration ng ating amino acids. So, blood specimen should be collected at the same time each day. Kasi, meron siyang diurnal variation. So, values are highest in the mid-afternoon and lowest in the early morning. So, dapat same time para hindi mag-vary sa specimen collection. General rules for amino acid analysis. So, in the plasma, it is required to have 6 to 8 hours fasting and avoid the effect of dietary proteins. So, kapag kasi mataas yung consumption mo ng proteins, pwedeng tumaas yung measure na amino acid. Kaya, magpa-fasting tayo. Collected in heparinized tubes. Separate cells from the plasma because they contain 100-fold increase in aspartic and glutamic acids. So, ito yung mga amino acid. Hemolysis should be avoided and deproteinization must be carried out within 30 minutes of a sample collection kasi magkakaroon ng interference yung ating proteins na yun. Kung baga tataas yung ating measurement nun. Kaya kailangan tanggalin. In the urine, kapag urine naman yung i-measure, random specimens is suitable. So, anong pinagkaiba ng ating random tsaka ng 24-hour? So, kapag random, ito ay yung anytime collection. Ihi ka lang, collect ka lang ng urine, pwede na ipatest for amino acids. So, tawag natin ng random amino acids or random testing ng specific amino acid. For quantification purposes or yung bibilangin na natin siya within the 24-hour, urine is preserved with thymol or organic solvent is required. So, 24-hour collection ng urine, lalagyan ng preservative para hindi siya mapanis or masira. Clinical significance. So, amino acids are generally used to monitor inborn errors of metabolism. So, usually pang mga baby unless kidney problems na. So, amino acidopathies. So, inherited disorders of amino acid metabolism. So, inherited, mga baby pa to. Alkaptonuria. So, absence of homogeneous state oxidase in the thyroxine pathway. So, tandaan natin itong mga to. Absence of homogeneous oxidase. Ito yung mga usual na natatanong sa ating mga exam and board examinations. So, tyrosine pathway which leads in the accumulation of homogenesic acid. So, clinical features, anorchosis or tissue pigmentation and darkening of urine upon standing at room temperature. So, pwede natin mga tandaan yan. Ano yung sakit? Alkaptonuria. Ano yung enzyme? Nakulang siya. Ano yung kanyang clinical features yun. So, laging tatandaan yan. Homocystinuria, impaired activity of cystathionin B synthase. So, results in elevated levels of homocysteine and methionine in blood and urine. So, clinical features, physical defects, thrombosis, osteoporosis, lens abnormality, and mental retardation. So, ito yung mga ating makikita sa ating homocystinuria. So, screening tests, pwede natin gawin. Modified Guthrie tests. So, used for blood yung ating specimen na ginagamit. Procedure. Blood specimen is placed on a filter paper which is then placed on agar plates with a strain of Bacillus subtilis. So, if gusto nyo makita yung in-depth procedures na to, hindi na natin kasi ito masyadong ginagawa or hindi natin ito ginagawa sa ating laboratory. Usually ito ay sa ating NBSP. Newborn Screening NBS. So, usually hanggang dito lang tayo sa collection. Blood specimen is placed on a filter paper. Hanggang doon lang yung ating ginagawa pag ganitong mga newborn screening. Tapos, sinesenda natin siya sa ating mga reference laboratories for the newborn screening. L-methionine sulfoxamine as antagonist. So, increased plasma levels of methionine will allow bacterial growth by suppressing the antagonistic effect of L-methionine sulfoxamine. So, para siyang suppressant do sa ating inhibitant. So, pagdating sa bakte, makikita nyo naman yan. So, screening test again, cyanide nitroprusside test. Cyanide nitroprusside test is added to urine. So, high levels of homocysteine will be detected by the development of purple-red color. So, positive result ng cyanide nitroprusside test is purple-red color. Has positive interference with cysteine. So, eliminated by adding silver. Reduces homocysteine but not cysteine. Kasi yung cysteine, nagkaka-interference siya. Homocysteine then reacts with nitroprusside to produce reddish color. MSUD or maple syrup urine disease. So, ito yung amoy maple syrup daw ang ating or ang urine ng ating patient. So, complete absence or the reduction in the activity of branch-chain ketoacidic acid or decarboxylase enzyme, hence blocking the normal metabolism of leucine, isoleucine, and valine, which result in the accumulation of them or naiipon sila sa dugo, sa ihi, at sa CSF. So, maple syrup urine disease. Clinical features. Characteristic maple syrup or burnt sugar type of odor of urine, breath, and skin. So, may maple syrup daw siya. Failure to thrive or hindi nabubuhay. Muscular rigidity, mental retardation, and hypoglycemia. So, nababa ang sugar. Screening tests. So, modified GU3 test ulit. Ang gamit ay for azaleucine as antagonist, and confirmatory test is amino acid analysis by HPLC. So, ano yung ating HPLC? Anong meaning nun? So, high performance liquid chromatography. 4 mg per dl of leucine is indicative of MSUD. So, kapag ito yung result na nakuha mo sa HPLC, ibig sabihin positive or confirmed na siya for MSUD. Phenylketonuria. So, deficiency of phenylalanine hydroxylase. So, ito yung enzyme na wala ka. Phenylalanine hydroxylase. That catalyzes the conversion of phenylalanine into tyrosine. So, phenylpyruvic acid. So, substrate accumulated by phenylketonuria. So, ito yung naiipon naman. Phenylpyruvic acid, since wala ngang phenylalanine hydroxylase, hindi nakakapag-produce ng tyrosine. So, ito yung byproduct niya. Product of phenylalanine deamination, neurotoxic, excessive amounts may cause mental retardation ulit. So, give a characteristic musty odor to urine. So, ito din tandaan, ito yung usually na tinatanong. So, yung mga amoy ng urine, inborn errors of metabolism. So, usually, sa amoy ng urine, pwede mo siyang ma-hint or suggested, parang screening na din para malaman mo, oy, may problema pala tong patient na to. Screening tests again is GU3 bacterial inhibition tests. So, pag ganitong inhibition test, pag hindi tumubo yung bacteria, ibig sabihin positive yung test or yung screening test mo. Ibig sabihin positive siya pag walang tumubo. Spores of Bacillus subtilis are incorporated into agar plate containing an inhibitor B2 thane-alanine. A paper disc impregnated with dried blood sample is placed in the agar. If the blood phenylalanine level is high, it counteracts the antagonistic and the bacterial growths affected by the presence of antibiotics. So, kapag mataas yung blood levels ng phenylalanine, ibig sabihin, kukontrahin niya yung ating inhibitor. Tapos, tutubo siya doon sa ating plates o sa bacteria. So, hanap tayo ng video. Bigyan ko kayo ng video regarding sa mga ganitong inhibition test. Pero usually, hindi natin siya ginagawa sa ating mga laboratories. Mga sa ano na siya, diagnostic na. Sa mga special laboratories na. Yung sa newborn screening or sa screening tests na siya ginagawa. Next, microfluorometric assays. So, filter paper disc containing the sample is treated with trichloric acetic acid. So, the extract is then reacted with a mixture of ninhydrin, succinate, and leucine leucylalanine in the presence of copper tartrates. Microfluorometric assay. So fluorescence of the complex is measured using excitation emission wavelength at 360 and 530 nanometers respectively. Direct quantitation measurement of phenylalanine in the blood, stained filter discs. So usually yung mga filter paper lang, yung mga filter paper discs na kinokollect tuwing newborn screening yung ating mga samples. At yun yung mini-measure para makita if meron niyang mga problems. So not affected by the presence of antibiotics. So urine testing. So used for monitoring for dietary therapy. And urine is reacted with ferric chloride. If positive for phenylpyruvic acid, ibig sabihin green yung color niya. Tyrosinemia. So characterized by the deficiency in the following enzymes. So fumaryllo acetoacetic hydrolase. Ulit. Fumaryl acetoacetate hydrolase. So tyrosinemia 1. Tyrosine aminotransferase, tyrosinemia 2. 4-hydroxyphenylpyruvic acid oxidase, so tyrosinemia 3. So may tatlong classes yan. So depende kung anong enzyme yung kulang mo. Elevated tyrosine and tyrosine catabolites, so methionine and phenylhydropyruvic acid in the blood. So leads to liver damage. So tyrosinemia leads to liver damage. Last, cystinuria. So nuria, so urine siya. Defects in amino acid transport system rather than metabolic pathway, affecting renal system and other vital organs. So hindi na siya sa metabolic. Sa ano na siya, amino acid transport na. Genetic defect in renal absorption mechanism. So 20 to 30 fold, ibig sabihin times increase in urinary excretion of cysteine. Cysteine is insoluble and precipitates in kidney tubules.